Last time, we watched PriminB have an explosion in biodiversity thanks to the advent of active predation. Today, these new organisms will diversify into the deep sea and intertidal zones. But first, a small new organism challenges the balance of the ecosystem. While they may be overlooked due to being microscopic, the microtrophs are one of the most diverse groups on PriminB. Different clades of microtrophs adapt to different niches. One of these niches is consuming dead organic matter. One clade hyper-specialized for this lifestyle so much that it would create black colonies over carrion large enough to be seen by the unaided eye. At this point in time, over 5.5 billion years ago, the climate on Primin B was comparable to the Cambrian period on Earth. This clade used the higher temperature by using it as a catalyst to undergo binary fission quicker, allowing it to reproduce rapidly. This clade became so specialized for feeding on organic matter that it was capable of growing on living organisms. This could cause necrosis to living things as their tissues are metabolized by the microorganism. It could be spread to other organisms by physical contact or by exchange through the water. This microbe began to infect and kill other clades at an alarming rate. It is called Mavrosoma for its black body, and it appeared 5.505 billion years ago. Mavrosoma began to plague populations of larger organisms, causing heavy tolls to all populations. But the populations that were at the most risk were of clades that were already declining due to competition with more adapted clades. In the upper water column, polybrachids and ovibrachids passively filter feed. Ovibrachids, however, are more fit for the lifestyle than other polybrachid clades. This, combined with death caused by Mavrosoma, eventually drove the ancestral polybrachids to extinction. Clades derived from the diastomes were also heavily affected. While most were able to sustain a population, the ancestral diastome could not adapt better than the tubistomes, pentopsids, or vermiforms could, eventually bringing them to extinction as well. As this extinction event, which we will call the Mavrim extinction, continued, Neteromorphs also began to face extinction. Some clades of Neteromorphs survived, however, by using an R-type selection strategy. If you remember from last episode, R-type selection is a quantity over quality method of reproduction where the clade makes it by reproducing faster than it could be killed. This form of reproduction often selects for a smaller body size and shorter lifespan, which is true in this new clade. Species of this clade can fill a variety of niches, from eating the larvae of other organisms to feeding off the dead integument of others. Its smaller body size also enables this clade to swim much quicker. These surviving deuteromorphs are called tachypinids, and they appeared 5.503 billion years ago. Mavrosoma spread quickly through the planktonic araeosomes near the ocean's surface, and by extent, the filter feeders that feed on them. Polypinids don't have sophisticated stomachs that are likely to kill Mavrosoma like the filterworms do, so their populations may plummet, especially since they were in competition with the filterworms. Like the last Neteromorphs, polypinids adapted by pushing deeper into R-type selection. One clade began to reproduce at younger ages and reach a smaller maximum body size. They eventually became so small that their fins could not propel them against the ocean currents, making them functionally zooplankton. Their fins are still necessary to pass water over the gill surface, so the fins aren't considered vestigial. This clade of planktonic polypinids is called Infermopoda, and it became the last surviving clade of archaea polypinids 5.502 billion years ago. Not all environments were affected by Mavrosoma. On land, the growing populations of Protofolia were spreading rapidly. Eventually, the land began to become crowded with Protofolia, creating competition to get sunlight. One clade competed by growing taller than the others. To support its growing weight, this clade coated its fleshy bark with a layer of chitin, which allowed it to grow multiple meters in height. The sail-like leaves of Protofolia catch a lot of wind, which doesn't affect small clades. Large clades, however, can be derooted and knocked over by the wind. To combat this, the large clades grow branched patterns of leaves that let more wind pass around them. The gametangia on top of this clade grows into large spheres before popping to release its spores. This clade is called Ramaphyta, and it appeared 5.489 billion years ago. Due to the high population of photosynthesizers on land and the introduction of ramophytes, atmospheric carbon dioxide dropped and oxygen levels rose. This caused a reverse greenhouse effect that made the average atmospheric temperature drop. Mavrosomes used the high temperatures of PriminB to quickly reproduce, but as the temperatures dropped, their numbers began to dwindle until they went extinct 5.496 billion years ago. Most carrion eaters survived the Mavrum extinction. However, when feeding on corpses, some organisms unintentionally eat small magnesiodonts. This unsurprisingly kills most clades of magnesiodonts, but some clades adapted to survive in other organisms' digestive systems. Eventually, these clades adapted for parasitism. They remain in their host digestive system for as long as possible while feeding and reproducing. These clades are smaller, reaching from microscopic to a few centimeters at their maximum lengths. When their host organism dies, members of this clade spread to other organisms that feed on their host carrion. These parasitic magnesiodonts are called gastroverms, and they appeared 5.495 billion years ago. Magnesiodonts are not the only organisms to have parasitic clades. Larval pentopsids are able to feed off the dead integument of other organisms. One clade, however, underwent neoteny and lived on a host organism until it was time to reproduce. This clade never formed shells, but they began to feed on more than just dead integument. Eventually, they became able to burrow into the flesh of their host organism. This clade then specialized its two mouths into proboscis-like feeding appendages capable of boring into a host's vascular tissue. Anytime they are attached to a host with a circulatory system, they intake their host's blood through one mouth, digest it, then expel the waste through their other mouth back into the host circulatory system. This exoparasitic clade is called Acutobrachids, and it appeared 5.49 billion years ago. Symbiosis is when at least two organisms interact with each other for the benefit of at least one organism. Parasitism is an example of symbiosis, but there are other forms of symbiosis, such as mutualism. In mutualistic symbiosis, both organisms are benefited. One clade of tachypinids developed a mutualistic lifestyle with larger species. This clade actively looks for larger organisms that are host to Acutobrachids. They then feed on the Acutobrachids, which gives this clade a meal and helps other organisms get rid of parasites. This clade's underside flattened, and their mouth took on a sandpaper texture. These adaptations allow this clade to latch onto their host and pry Acutobrachids off of their host's integument. This clade of tachypinids is called Radostoma, and it appeared 5.487 billion years ago. Mavrosoma was not the only new microscopic clade of this time period. On Earth, some clades of bacteria use a mechanism called quorum sensing to regulate population density and gene expression. Depending on population density and other environmental factors, these clades of bacteria release chemicals called autoinducers, which other bacteria respond to. Tenax malia and its descendants regulate their different types of cells this way, and a clade of chemophagus has also been using quorum sensing. Like Earth's Vibrio like that one, this variant of chemophagus uses a chemical called luciferin as an autoinducer. Luciferin is a luminescent chemical that glows a yellow-green light when this clade releases it. The glow from this clade is not visible in thinly spread populations, but as the population density climbs, the glow grows brighter. This clade lives around hydrothermal vents all over PriminB, but it is especially noticeable in the deep sea. It is called chloroluxus for the color of light it emits, and it appeared 5.486 billion years ago. Marine snow, which is dead biomass that falls to the seafloor, is a large part of dorsostome diets. Most of the marine snow falls to the deep sea, where there aren't many organisms to feed on it. Given the lack of competition in the barren deep, some clades of dorsostomes were inclined to move deeper. As this clade moved deeper, however, the immense pressure began to crack and damage its shells. Sudden cracking of their shells is hazardous to dorsostomes because the broken pieces of shell can damage their soft tissues. This clade ultimately adapted to never develop a shell and leave their soft tissues exposed. Ancestral dorsostomes partially got their color from the pigment within the orange araeosomes that they eat, but this deeper clade does not have access to araeosomes, so it lost most of its pigmentation. These deep sea dorsostomes are called Malacoris, and they appeared 5.484 billion years ago. The deep seas are yet to house any predators. Some clades of vermiforms took advantage of this and migrated to the deep sea. However, their eyes help them much less in the deep sea. While chemoreception may help them find mates in the darkness, most mates of this clade meet after being attracted to the glow of large colonies of chloroluxus around hydrothermal vents. Over time, this transformed into a symbiosis between these vermiforms and chloroluxus. This clade of vermiforms collects colonies of chloroluxus between their segments. They feed these colonies by excreting a sulfur-rich waste product from special glands beneath their segments. They use the glow between their segments to attract mates where the brightness of hydrothermal vents are not available. This clade of glowing vermiforms is called Luxiverma, and it appeared 5.481 billion years ago. As a prey species moved to the deep sea, potential predators were inclined to follow. Luxiverms don't bury themselves in the detritus, since that would defeat the purpose of showing off their bright glow to mates. One clade of monodonts took advantage of this easy source of food. This clade developed a pair of tendrils around their mouths that assist it with picking up food. Early on, this clade found mates by being attracted to the glow of luxiverms. They improved upon this by producing their own bioluminescence. They use a different strain of luciferin that glows blue. However, the ancestors of both luxiverms and monodonts cannot differentiate green from other colors, which makes both of these clades susceptible to confusing each other as mates. This clade used this to their advantage. They lured luxiverms to them by using the glow of their tendrils to mimic luxiverm mates. These deep-sea monodonts are called Mendeluxus, or false light. They appeared 5.479 billion years ago. One clade of Curvatesta also specialized for the deep. Like Malacoris, their shell was a potential hazard. Unlike Malacoris, however, this clade softened its shell and internalized it under its integument. This allows them to still reap the benefits of having a shell without the risk of it cracking under the deep sea pressures. Since clades derived from pentopsids are able to differentiate green light from other colors, it is able to recognize the blue glow of Mendeluxus and pursue it. It is able to attack, almost unseen, as it does not produce any light of its own. In the event that it cannot find any Mendeluxus, it patrols the seafloor for Malacoris. This clade of curvitests are called Osteothalamus, and they appeared 5.476 billion years ago. Some deep-sea vermiforms did not gain bioluminescence like their relatives, but instead adapted to feed on the compounds spewing from hydrothermal vents. Through mutualistic symbiosis, this clade cultures colonies of chemophagus in their digestive tracts. These chemophagous colonies help them turn compounds released from the hydrothermal vents into energy. In return, the chemophagus use this clade's digestive tract as a stable environment to grow in. Since this clade rarely has to travel, it reverted to a sessile lifestyle. Instead of moving like other vermiform clades, this clade roots its posterior end of the soil around hydrothermal vents and remains there for the rest of its life. It inhales the compounds that are released by waving specialized appendages in its mouth. Since vision no longer offers great advantage, this clade's eyes become a vestigial organ. The eye is reduced in size and it lost its ability to detect light. These organisms often live in clusters of 20 to 30 and reproduce by spewing their gametes into the water. When their gametes meet, they produce a microscopic larva that quickly settles near the seafloor. This sessile vermiform clade is called Ventostoma, and it appeared 5.475 billion years ago. Back in shallow waters, gravitational pull from PriminB's moon creates rising and lowering tides. The area affected by these tides is called the intertidal zone. Many organisms live within the intertidal zone, including some tubistomes. If the water recedes, it may leave the tubistomes prone to drying out. To prevent this, one clade developed thicker integument that retains water better. They also developed glands around the perimeter of their shells that secretes a thick mucus that allows them to lock in moisture. During low tide, buoyancy is no longer helping lift these tubistome shells. This encouraged this clade to have smaller, more lightweight shells. Their muscular foot also grew much stronger, allowing them to better traverse land. This clade is adapted enough for land that it can occasionally leave the water for short periods of time to eat land flora. These early land tubistomes are called pulvinopods, and they appeared 5.464 billion years ago. Some pentopsids also inhabit the intertidal zone. Like pulvinopods, the intertidal pentopsids developed lighter shells. Ancestral pentopsids did not move very actively, relying on the current to do most of the movement for them while using their arms to supplement movement. But since there are no strong currents in tide pools or on dry land, these pentopsids have to rely on using their arms more actively. Active movement like this selected for a more bilaterally symmetrical body style. All 5 of their arms are shifted towards one side, with the inner 3 arms being used for primary movement while the outer 2 arms for its primary vision. The eyes on this clade's movement arms moved closer to the organism's center mass, and the eyes on the visual arms are larger and raised. When they find a mate, they dig nests into the mud and spawn in these nests. These land pentopsids are called sumopsids, and they appeared 5.46 billion years ago. With its prey species beginning to spread to land, Testaruptus was inclined to follow. One clade of Testaruptus grew stronger walking limbs and a broadened stance that allowed them to walk on land for limited periods of time. Aquatic Testaruptus have tissues between their segments that act as a gill surface, but this clade internalized these tissues. Each section of tissue eventually developed into a small lung-like organ between each segment. Small muscles that line these lung-like organs allowed them to draw in oxygen and diffuse it into the organism's hemocoel. The ancestral Testaruptus fins that pushed water over the gill surface became vestigial and reduced greatly in size. This organism spends most of its time in shallow waters and emerges to hunt for pulvinopods and sumopsids. These land hunters are called latopods, and they began hunting 5.455 billion years ago. As organisms began to populate the land, they provided plenty of organic matter to form detritus. This led a clade of vermiforms inhabit land soil. This clade must stay wet in order to breathe, so it only exists on beaches and other wet environments. This clade is thinner and softer than other vermiforms, similar to earth annelids. This clade is called Detritifagous, and it began breaking down the nutrient-rich soil 5.45 billion years ago. As all of these organisms began moving to land, they brought parasitic gastroverms with them. But in order to maintain a population, the gastroverms had to adapt to these new conditions. One clade grew much smaller and was much less harmful than its ancestral clade. By keeping its host alive longer, this clade was able to reproduce for a longer period of time without having to find a new host. Instead of solely spreading through the carrion of dead hosts, this clade can also spread through their host's excrement. These more sophisticated parasites are called vastiverms, and they appeared 5.447 billion years ago. One new clade managed to move to land with great difficulty. This began with a clade of Platyventra that fed on their new prey options in shallow water. A basic skeleton made of tissues similar to cartilage helped this clade retain rigidity out of the water, but it had difficulty moving when on land. Ancestral Platyventra moved by flapping the fin on the underside of their body, like earth rays. This clade altered this by having thicker cartilaginous tissues in some areas of their fins, creating 4 simple protolimbs. If this clade were to get washed up onto land, it would drag itself back to the ocean using these limbs. This clade hunts by hiding itself in the sand around beaches, where it waits for other organisms to return in the water to either rehydrate or reproduce. When prey gets in range, it ejects itself from the sand using its limbs. These organisms are called paraxapods, and they were the last organism to appear in the Mavram period, as it ended 5.44 billion years ago. During this episode, we watched 19 new clades emerge as life began to expand into new environments. This will continue next time, as life begins to live on land permanently and the sea creatures move into freshwater. If you would like to know more about PriminB, make art that can be displayed in these videos, or get insights on future episodes, make sure to join our Discord server. Link is in the description.